Hello there. So in the last video we have already started with cellular respiration topic, and in the last video itself we have talked about the basic of cellular respiration, respiratory substrate, the types of respiration, that is aerobic and anaerobic. Even we have seen the differences. So today I'm going to move ahead with the next subtopic, that is glycolysis, the process of glycolysis. And as the name suggesting, that glycolysis process is something that is glucose lysis. Glyco means glucose, lysis means splitting. So here splitting of glucose will be taking place. So I can say, can I write here that in this case glycolysis, in the process of glycolysis, one molecule of glucose, it is going to get converted into two molecules of pyruvic acid. So glucose is a 6 carbon. It is a 6 carbon compound which is going to get converted into two molecules of pyruvic acid having 3 carbon in each molecule of pyruvic acid. Right? So glycolysis, during glycolysis process, what is going to happen? One molecule of glucose is going to convert it into two molecules of pyruvic acid. But it is not as simple as it looks. Okay, it is not a single step process. It is going to be a very complex process. It is around 10 this whole process is going to take place in around 10 steps that we will see very soon. But yes, it is a complex process. Now another thing that I can tell you here, that even we discussed in the last video itself, that glycolysis is the common pathway for both the type of respiration. Common pathway for both the type of respiration, that is aerobic and as well as anaerobic respiration. So whether the respiration is aerobic or anaerobic type of respiration, the glycolysis process is going to occur. Okay, and this process is also known as EMP pathway. Why? Because it was given by three scientists, namely Amberton, Merhoff, and Parnas. So these were the scientists who gave this glycolysis pathway, and for which they have also been awarded Nobel Prize. So that is why this process is also known as EMP pathway. Okay. Now, what is the site of glycolysis? The site for glycolysis is cytoplasm. It occurs in cytoplasm of the cell. Now, let us start with the glycolysis process, and as I have told you that it is not a single step process. It is going to occur in around 10 steps. So let us start. Glycolysis process. So glycolysis process it's a process in which glucose is going to get converted into pyruvic acid. So let us start with glucose only. So here one molecule of glucose, which is having 6 carbon in it, is going to get converted into something known as glucose 6 phosphate. So here what is happening? On the 6 carbon of this glucose, a phosphate group is being added. So where does this phosphate group comes from? It must be coming from the breakdown of ATP into ADP, and whatever phosphate is being released here, it has actually get joined on this glucose, and This process is happening in the presence of an enzyme known as hexokinase enzyme. Okay, so the in whole process you will see that whenever I will be releasing breaking down ATP, or ADP is forming, or ADP is getting converted into ATP, we will see this kinase enzyme will be used here. And also, since glucose is a 6 carbon molecule, that is why the word hexokinase. Okay, and in this process it requires magnesium. So magnesium should be present. Also, since I can say here the glucose is being added with phosphate, so here phosphorylation is happening. Since that is why the name of this process is phosphorylation. This name of this very step is phosphorylation. Now glucose 6-phosphate is also a 6 carbon compound, and since one molecule of glucose is getting converted here, so here also we are getting one molecule of glucose 6-phosphate. Okay, now second step, that this glucose 6-phosphate is going to get converted into one molecule of fructose 6-phosphate. Okay, again here in this step we will be requiring magnesium, and also since fructose it is the isomer of the glucose 6-phosphate glucose. Fructose is of isomer of glucose, so the name of the process is isomerization. And the enzyme that is involved here is basically, since isomerization is happening, so isomerase enzyme. And since the substrate was glucose 6-phosphate, so I can write it is, the enzyme name particularly is phosphoglucoisomerase. Okay, now one molecule in third step, one molecule of fructose 6-phosphate, it is going to get converted into fructose 1,6-diphosphate. Already in the case of fructose 6-phosphate, there was phosphate group present on the 6th carbon. In this, again a phosphate group is being added in first carbon, on the first carbon as well. So one molecules of fructose 1,6-diphosphate is being formed. Here also 6 carbon present, here also 6 carbon present. Okay, and since again the phosphorylation is happening, a phosphate group is being added. So from where does this phosphate group must be coming? It must be coming the conversion of ATP into ADP, right? And whatever phosphate is being released from here, it is being added on fructose 1,6-diphosphate. Okay, for this process, since phosphorylation is happening, this step is also known as phosphorylation process. And the enzyme that is being used here, it is, since ATP is getting converted into ADP, and I told you wherever in this respiration processes, whether it is glycolysis, Krebs cycle, we will see wherever there is conversion of ATP into ADP or ADP into ATP, the kinase enzyme is going to get utilized. So kinase enzyme, which kinase enzyme? Since here fructose 6-phosphate was present, so the enzyme will be phosphofructokinase enzyme. Okay, so we have got one molecule of fructose 1,6-diphosphate here. Now the next step is this fructose 1,6-diphosphate. It is going to get split up. Into two different compounds, namely 3-phosphoglycerol glyceraldehyde, also known as 3-PGAL, and it is a 3-carbon compound. So 1 molecule of 3-phosphoglyceraldehyde and also 1 molecule of something known as dihydroxy acetone phosphate. Dihydroxy acetone phosphate, basically it is known as DHAP, and it is also a 3-carbon molecule. Since 6-carbon molecule is getting converted into 3-carbon, 2 molecules which is having 3 carbon each. One is 3-phosphoglyceraldehyde and one is dihydroxy acetone phosphate. And this is happening in the presence of an enzyme, that is aldolase enzyme. And since here the fructose 1,6-diphosphate is being split up, this process name is known as simply lysis. Lysis means breakdown, splitting up, right? Now, the fifth step will be, since these 3-PGL and DHAP, these are isomers of each other, and with DHAP the glycolysis process cannot be continued. So this DHAP is ultimately going to get converted into 3-PGAL. So this is going to get converted into 3-PGAL in the presence of enzyme. Since isomerization is happening, so enzyme must be an isomerase enzyme. Which isomerase enzyme? Phosphotriose isomerase enzyme. So ultimately now we have got 2 molecules of 3-PGL, right? And since isomerization is happening, so the name of this step is isomerization. So basically these are the first 5 steps of glycolysis process: 1, 2, 3, 4th and the 5th step. And here, since we have utilized the energy, instead of being production of energy, the energy is being utilized. That is why this phase is also known as energy investment phase of glycolysis. These first five steps are known as energy investment phase of glycolysis, also known as preparatory phase. Okay, so by the end of 5th step, we have got 2 molecules of 3-phosphoglyceraldehyde. So by the end of 5th step, we have already got 2 molecules of 3-phosphoglyceraldehyde, PGAL, which is a 3-carbon compound. Now, this 3-phosphoglyceraldehyde in the 6th step is going to get converted into 1,3-diphosphoglyceric acid. Now basically this step is a 2-step process. So what is happening? That in the very first step, that is your 3-PGAL, it is getting converted into 1,3-diphosphoglyceraldehyde, and since here one phosphate group is being added up, so that phosphate group is coming from phosphoric acid. So this is releasing inorganic phosphate which is being added here, and 3-PGL is being converted into 1,3-diphosphoglyceraldehyde. Now again this 1,3-diphosphoglyceraldehyde is going to lose a proton, and whenever there is a proton is being released, oxidation is happening, and that removed proton will be ultimately accepted by NAD+, because this is the coenzyme, and it is going to get converted into NADH+, H+. That is your reduced coenzyme, and ultimately you are going to get 1,3-diphosphogylic acid, which is present here, right? So I can write here that 2 molecules of 3-PGL are getting converted into 2 molecules of 1,3-diphosphoglyceric acid. It is also a 3-carbon compound. And what is all happening here? Let us write it, that a phosphate group is being added, which is coming from phosphoric acid, right? And also this step, since 2 molecules are present here, so 2 molecules of phosphoric acid and also 2 molecules of NAD+ are getting converted into 2 molecules of NADH+, H+. And this step, since it is required, in this step both the phosphorylation and oxidation is happening, this step is known as oxidative phosphorylation. Oxidative phosphorylation. And this step is going to happen in the presence of enzyme phosphoglycerodehydrogenase. Dehydrogenase enzyme. Okay. Now, in the very 7th step, this 1,3-diphosphoglyceric acid is going to get converted into 3-phosphoglyceric acid. 3-phosphoglyceric acid, again a 3-carbon molecule, and since 2 molecules were there, so here also 2 molecules of 3-phosphoglyceric acid are being produced. Here what is happening? One phosphate which was added in this diphosphoglyceric acid, it is being eliminated, it's being removed from this phosphoglyceric acid. So that phosphate group must be added up somewhere, and where it must be added? It is being added into ADP, and 2 molecules of ATP ultimately will going to get converted into 2 molecules of ATP with the addition of this phosphate group. Okay, and again ADP is happening here. So enzyme will be kinase enzyme. Which kinase enzyme? Phosphoglyceric acid was there, so phosphoglycerokinase enzyme. Okay, and since a phosphate group is being removed from this molecule and being added to this ADP, so here the step is dephosphorylation. Dephosphorylation, right? Now in the next step, what is going to happen, that this 3-phosphoglyceric acid is going to get converted into 2-phosphoglyceric acid, again a 3-carbon molecule, and since 2 molecules were there, here also 2 molecules of 2-phosphoglyceric acid is being produced. So what is happening? Earlier the phosphate group was present on the 3rd carbon, now it is localized to, or it has changed the location and it has shifted to the 2nd carbon of the phosphoglyceric acid. So here kind of change is happening, and the change is related to mutation. So the enzyme that is involved is mutase enzyme. Which enzyme? Mutase enzyme. Okay, and this step is known as, since these are isomers of each other, this step is known as isomerization step. Okay, and now once this 2-phosphoglyceric acid has been formed, it is again going to get converted in something known as 2-PEPA, that is your phosphoenol pyruvic acid. Phosphoenol pyruvic acid, again a 3-carbon molecule. Since 2 molecules were there, 2 molecules of this PEPA is being produced, phosphoenol pyruvic acid. Okay, and this is happening in the presence of which enzyme? Enzyme enolase. Enzyme enolase, and this enolase enzyme works by removing water. So here 2 molecules of water are being removed. Since water is being removed, the step will be known as dehydration step. Addition is hydration, removal is dehydration. Okay. Also, this step requires magnesium here. I will just mention magnesium also. This step requires magnesium as well. Okay. Now, this 2-PEPA is going to get converted ultimately into 2 molecules of 2-phosphoenol pyruvic acid is going to get converted into 2 molecules of pyruvic acid. Again, a 3-carbon compound. Okay, and here in this step, what is happening? 2 molecules of ADP are going to get converted into 2 molecules of ATP in the presence of magnesium plus potassium ions, and this is happening in the presence of an enzyme known as pyruvic kinase enzyme. Okay, and again here phosphate group is being removed from this 2-phosphoenol pyruvic acid. So the step name is dephosphorylation. So we have ultimately got 2 molecules of pyruvic acid, which was the goal of glycolysis process. If you remember, we have started with that, we have said that 1 molecule of glucose, it is going to get converted into 2 molecules of pyruvic acid. So we have started with 1 molecule of glucose and we have got now 2 molecules of pyruvic acid. So in this phase of glycolysis, like if I talk about the 6th step, 7th step, 8th step, 9th step and 10th step, all these 5 steps from 6 to 9 steps, they are known as, since energy is being produced here, so this phase is known as energy generation phase or simply the payoff phase. Okay, so this was all about glycolysis. Also, one thing that if you will just look at the whole process of glycolysis, you can see here we have produced 2 molecules of ATP and here 2 molecules of ATP are being produced. So total ATP that are being produced is 4 ATP. But in the first 5 steps, we have seen that 2 molecules of ATP are also being utilized here and they have converted into ATP, right? So 4 ATPs are being produced, 2 we have already been utilized. So net gain of ATP will be only 2 ATP, 4 minus those 2 that we were utilizing, and ultimately you have got net gain of ATP, that is 2 ATP. Also, from this process we have got 2 molecules of NADH as well, which will further enter into electron transport chain and get converted into ATP. We will see how. So stay tuned for my next video, and in that we are going to start with another things about respiration. We are going to talk about anaerobic respiration, and then we will talk about Krebs cycle and etc. that are the part of aerobic respiration. So until then, stay safe, and I'll see you in the next video. Bye bye. Take care.